Every time your heart beats, trillions of tiny molecules fire into action. But the true star of this process isn't your heart muscle itself. It's a tiny familiar ion called calcium. Today we're going to zoom in on the molecular level to see how calcium ions, troponin, and tropomyosin work together to turn an electrical signal into physical movement. Then we will take a look at peer-reviewed research displaying what troponin actually does when calcium arrives. Every skeletal muscle in your body contains many fascicles. These fascicles contain many muscle fibers, which contain many myofibrils. Each individual myofibril contains a sarcomere. This is where muscle contraction happens. Muscle contraction can be explained by the sliding filament theory. This theory expresses the interaction between actin, a thin filament, and myosin, a thick filament. When myosin pulls on actin, the muscle shortens and contracts. But here's the catch. Myosin can only grab actin when the binding sites are exposed, and normally those sites are physically blocked by tropomyosin, kind of like a safety bar. In order for this process to occur, calcium ions are released from the sarcoplasmic reticulum as a muscle cell is excited. These calcium ions bind to troponin C, which acts like a molecular switch. Troponin changes shape, allowing tropomyosin to move out of the way and enabling myosin to access actin. Now, cross bridges can form. ATP is used and the muscle contracts. When calcium is removed or reduced, tropomyosin slides back and the muscle relaxes. To understand what really happens inside the thin filament, researchers Sun, Liu, and Irving studied demembranated heart fibers, known as trabeculae, from rats. Researchers replaced the rat's native troponin C with a fluorescent variant of troponin C to monitor its motion of troponin helices throughout muscle contraction. Here's what they discovered. Calcium binding alone caused large structural changes in troponin. Even when myosin was blocked with blebistatin, activation still occurred. The thin filament, actin, doesn't need myosin to activate it. Myosin inhibition with the use of blebistatin did not reduce this activity, indicating that this activation of actin is intrinsic to thin filaments and not dependent on the binding of myosin. This determines that calcium binding to troponin is the main driver of cross-bridge formation and thus muscle contraction. You may be asking, why does any of this matter? This is because in real life, small changes in this system can cause major disease. Marston and Zamora showed that mutations in troponin can distort how it responds to calcium. Abnormalities in calcium ion sensitivity or the structure of troponin can lead to cardiomyopathies, where heart muscle becomes weak or overly stiff, and muscle weakness. So understanding this mechanism isn't just cool, it's medically important. We can connect this concept to much of our course content, like cell signaling, since calcium ions act as second messengers, allowing electrical signals to create a mechanical contraction, and protein structure and function, since calcium ions can change the composition of troponin, allowing tropomyosin to shift and allow an actin to myosin interaction. For cell structure, actin and myosin allow motor functions within the cytoskeleton, and in cellular respiration, myosin heads use ATP hydrolysis to power muscle contraction. So here's the big picture. Calcium ions act like tiny messengers. They bind to troponin, troponin changes shape, and tropomyosin moves. Myosin can now pull actin, and the muscle contracts. Microscopic choreography is happening right now in every heartbeat and every movement you make. And thanks to research, we now understand the exact molecular switch that makes it all happen.